Why do viruses mostly affect only one species? The key has to fit the lock. Unfortunately for us, there are very few master key viruses floating around out there. Viruses often recognize species-specific cell surface proteins that they then use to enter the cell. These proteins may have large or small differences in their amino acid sequence and function, and some proteins may be species-specific. Very few master key viruses. It's theoretically possible to have a single master key virus that fits all cell surface proteins. Or is the variation too great? It's not a virus. But one of the most broadly successful pathogens in this regard is Toxoplasma gondii. It's obligate intracellular, meaning it can only live inside other cells. Similar to a virus, but it's actually a single-celled eukaryotic parasite. T. gondii cells are organized and separate their DNA into a nucleus. Like animals, it can infect basically all warm-blooded animals and many different cell types within those animals, but it only sexually reproduces in cats. How T. gondii manages to infect such a broad range of animals is still being studied, but I saw a talk recently by Drive John Boothroyd on the topic, and the very short answer is that it brings all of its own receptors, injects them into the host cell, and then uses them to enter the cell, which is very cool. What about rabies? The rabies virus also infects primarily warm-blooded mammals, but has been shown in labs to be capable of infecting reptile and bird cell cultures. It is almost never found in rodents, however. It's because a lot of the machinery is not universal, especially the parts the virus used to get into cells, then into the nucleus, and then to use the DNA there to make more virus. Those parts are generally very specific to each species, and even to types of cells within a species. To elaborate on your correct answer, cellular machinery is universally very similar. But in between two individuals within a species you see enough small variation in proteins and their structures to cause differences in outcomes when it comes to infection and other diseases. Between two different species, the variations stack up. This is my area of expertise. Let's first address the most of the machinery of cells is universal statement. While this is true in a sort of surface understanding, that eukaryotes share many basic fundamental processes, and these processes are carried out by related proteins, there are many details that differ at the smaller scales. At the near atomic scale, we will find that organisms will have variations in amino acid sequence that lead to slightly different properties. The main changes I'll address here are changes to amino acid residues at the surface of a protein, and changes that alter co- and or post-translational modifications. If an amino acid on the surface is required for a binding event, and we change that residue, we will change the characteristics of that binding interface. The molecular details there will be different, charge may be altered, geometry may change. A bulkier residue could cause steric clashes. Even a mutation away from the binding interface, alastery, may change the properties of the interface. What once was a high affinity interaction may now be inhibited. Changing a single amino acid can abolish binding interactions. This can restrict a virus to a particular host with a particular interface. If one or a few residues can reduce binding, imagine insertions or deletions of large chunks of protein accumulated over the course of evolution or intron/X and splicing. Another source of variation in proteins are modifications that enzymes in the cell add to proteins. There are enzymes within cells responsible for trimming peptides, adding highly charged groups, adding sugars, and even adding smaller proteins to existing proteins. Let's consider an enzyme that catalyzes the addition of sugar molecules. This is called glycosylation. The complement of enzymes that play a role in this process are slightly different species to species and even tissue to tissue. The infamous bird flu or swine flu are an important example related to the process of glycosylation. The receptor for influenza virus hemagglutinin, HA, its surface protein, is a sugar, sialic acid that is connected to some other sugars and a cell surface protein. The sialic acid binding site for HA has different affinities for different configurations and linkages of these sugars. A flu virus that mainly interacts with birds will have a binding site optimized for bird receptors. A virus that mainly infects humans will have slightly different binding site that is optimized to bind human receptors. Avian influenza virus will prefer the alpha-2, 3-linked sialic acid. When human cells glycosylate their surface proteins, they end up making alpha-2, 6 linkages for sialic acid. So naturally human influenza viruses will have a binding site optimized for those. Unfortunately pigs, turkeys, and pheasants have both. Influenza viruses that have accumulated mutations in their binding site may be at an advantage in those hosts, leading to more virus with higher affinity for alpha-2, 6 bearing receptors. At the next step up in scale, protein dynamics are another largely unexplored area in protein variation. The extent or rate of how quickly a protein is moving or breathing may alter binding interactions. If an interface is hardly ever exposed in one protein due to a difference in how flexible or inflexible that protein is, then the affinity may be reduced. Again, this is unexplored territory for the most part. Most of the work in this area so far has been related to antibodies or therapeutic targets. Another scale to consider is the amount of a specific protein in a given cell or cell type. Viruses replicate by hijacking their host's cellular machinery, using the host's energy, building blocks, organization, and architecture as a virus factory. Every single protein in a virus is highly evolved and specialized to particular environment, meaning pH, temperature, available molecules, and host proteins. Many viral proteins carry out multiple functions, what I would call genetic economy, and so rely on the presence of multiple host proteins at certain levels at specific points in a virus replication cycle for optimal replication. There is cell-to-cell variation in the amount of specific proteins. This variation could be due to the tissue type. Consider the complement of protein in a muscle cell vs a neuron, or even just different developmental stages of growth. Comparing one organism to another organism will show incredible variation in levels of most every protein. This is mainly why viruses are limited to infecting only certain tissues. Sure, all cells share the same DNA code, but levels of RNA and protein are way different. If one is restricted to T cells, because its envelope surface protein binds to 2 proteins that are only expressed in helper T cells, it doesn't go about infecting your airway epithelial cells. There are also differences in immune systems. This is a huge field that I can't possibly cover. But briefly, one example. In many cells, there are immune functions that can restrict a virus from replicating egg by recognizing virus DNA, RNA, lipids, sugars, or proteins that activate responses that prevent the virus from replicating or spreading. Not all organisms have these functions. So really, machinery of cells isn't all that universal, and viruses are very compact and have only one highly specialized purpose: to replicate itself. Viruses only carry a few of their own proteins, typically a dozen or so, and heavily rely on their viral proteins forming contacts with specific cellular components, and these interactions are largely dependent on very small, sub-nanometer to tens of nanometer, binding interfaces. Change in either the virus or the host may have potential to lead to increasing the binding affinity, paving the way for this jumping from one species to another. But it has to be a perfect storm of accumulated mutations. Edit a coronavirus specific example. The coronavirus spike protein is synthesized as a precursor that requires proteolytic processing at a certain amino acid sequence before it becomes activated. Without the specific host proteases that recognize that sequence in the right place at the right time, the virus spike protein cannot become fusion competent, trigger, and allow entry into the cell. Thanks for this informed answer. Fascinating. You spend most of your answer on binding on the surface of a cell, and also many other answers do. May I ask why binding is so hard for a cell surface? We have glue that works for nearly any surface. Did cells make it extra hard to attach to them as a defense mechanism? Viruses exploit cellular machinery, and this machinery is different in different species. There are differences in glycosylation, protein expression, protein conformation, lipid and sugar metabolism, protein expression, innate and adaptive immune response, etc. they don't need to specialize. In fact, the opposite is true. There's so much diversity out there that any random host will do. They're incidentally matched to different species. Not purposefully. It does take many generations to adapt, because the virus follows the host's lifestyle, so obviously it would be a waste to have adapted very well to humans and then shift to something very different. Adapting to a host can be extremely successful. Trying to work for multiple would be a waste. The cell machinery may be the same but different species response, reproduction, environment and intricate interactions aren't. The main action is not so much the virus finally being in the cell, it's how it got there in the first place and how it got into the host body in the first place. That's what governs its success, and that's what is extremely different between species. The simple answer is that the cells in each species are incredibly complexly unique, and in order to counter this and break in, viruses have to specialize their attack strategy to the point that it's ineffective against anything else. Viruses have very simple genomes that often only code for a few proteins. That includes the machinery to produce and assemble more of itself, and some structural proteins that help anchor virions to cells or evade the immune system. Protein interactions are normally very specific and very precise, and the odds are pretty low that a protein interaction in one species will work as well in another. As an interesting side note, plagues usually comes from animals and are a mistake from the perspective of the virus. It kills humans far too fast to be useful because it was designed to live for a long time in, for example, a cow. Some viri can't live at all in other species. Some act differently. To add to what others have said, many viruses co-evolve with their hosts. The most successful viruses are able to keep a host sick and infective for a long time without causing mortality, since a dead host is less likely to infect others. There are cases where these very specialized viruses encounter species that are similar enough to their natural hosts that they can still cause illness, but with a much higher mortality rate. For example, Macassine alpha herpes virus 1 typically infects macaques and causes illness similar to human herpes viruses if Macassine alpha herpes virus 1 infects a human. However, it can lead to severe neurologic dysfunction and death. Same goes the other way around. Human herpes infecting other primates. Basically, it's like a program made for a specific operating system. So for instance, macOS from Apple. If you try to run it on Windows, it doesn't work because the program requires specific features that only the specific operating system can provide. A virus needs specific features of an organism to survive, and if it can't find that, it can't keep itself alive or even work. That's the most basic way I can explain it. Also, there's no point in a virus needing to affect multiple species, because, generally speaking, a species will mostly be in contact with their own species. Also, there's no point in a virus needing to affect multiple species, because, generally speaking, a species will mostly be in contact with their own species. Not necessarily. In nature, cats, for example, come into contact with rodents and birds a lot more often than they come into contact with other cats. I'd say that probably holds true for a lot of species that exist as solitary predator animals. It's so they get a proper host. Each species is different and the virus is specialized to get the best host for that virus. They need to be able to detect what cell they are infecting, so they don't just infect some bacteria in your body and die off when your immune system finds the bacteria. Viruses are a product of their environment. If you look for bacteria in the soil outside your home, you can bet good money that there will be a bacteriophage or 3 in that soil that specifically targets the bacteria there. So unless the environment you evolved in encompasses the entire universe, you won't get a universal virus. I feel like nobody gave the right answer yet. Virus can go to other species, but when this happens they usually have problems with spreading and don't become kind of a big deal to the new hosting population. Let me be more detailed. Viruses need to make the host sick to the point of it spreading them, but not too sick to the point of killing the host. If they kill the host, they are also killed. If they don't make him sick enough, they will be killed by his immune system. So they have to make him sick in the right amount. If some pig's virus starts to spread in a human population, For example, it will be too strong for humans because we have a worse immune system, ending up killing us more than spreading the virus. If the same virus goes to a horse, nothing will happen to him because soon his immune system will kill it. That does not fully make sense to me, wouldn't? In that scenario, big viruses would regularly kill people, plus the viruses of billions other species. He's saying that usually a virus that wouldn't really bother an animal would kill a human, and if everyone who gets it dies, it's harder to spread to an entire population. I'm not entirely sure this is accurate, as every few years a new virus will jump species from an animal, usually farm animals or bats, and they usually have higher mortality than common viruses, but not to the extent that they would burn themselves out. Examples being bird flu, swine flu, SARS, and MERS. I get it that there is no advantage for a big virus to quickly kill a human. But if it were not somehow hard, there would also be no disadvantage to it. It is not worse than the virus dropping on the floor and dying there. Hey, thanks for watching, and don't forget to subscribe.